So outline, this is what we will cover in our lecture. So, okay, the first one will be the definition of biotech itself. And then we go on to the fields in biotechnology. Kita akan tengok the different fields involved in biotechnology. Dia dah berkembang sampai ke mana in this sampai ke current situation. And then we have the omics technology. All the omics tu, genomics, proteomics, metabolomics, all that. Okay, kita akan tengok sikit-sikit. And then we'll go into the techniques in biotechnology and also the biotechnology products. Okay. Okay, so the learning outcome to define and differentiate the various fields in biotech. Kita ada few fields in biotech, so you have to define and know what the difference between each field tu. And then to explain the several main techniques in biotechnology, we have DNA cloning, we have recombinant DNA technology, we have gene editing and gene therapy. This one you have to know the differences and apa satu-satu tu punya advantages or disadvantage. Okay, you can read more on that. Tapi kalau soalan exam tu dia akan bagi situation lah, kalau macam ni dia bagi situation scientist ke apa-apa ke, you tengok dia buat macam mana tu, how apa yang you boleh apply. Dia tengah nak buat cloning ke, dia nak buat recombinant ke, something like that lah. Okay. Alright, soalan number 3 is to list several biotechnology application and also the products. Okay, first thing first, what is the definition of biotech? Mak, saya takut ni lah. Internet saya kejap ya. Okay, alright. So definition of biotechnology. Any technological application that uses the natural biological system to make or modify product for a specific use. Selalunya memang very broad dia punya term sebab dia melibatkan banyak sangat field. Okay, tapi this biotechnology has made impact. Okay, banyak dia punya benefits dah yang kita dah nampak sebelum ni. Dia selalunya yang paling kita nampak ialah GMO, which is genetically modified organism. Contoh macam tomato ni. This is a normal tomato. Tomato kalau you tengok ada patches sebenarnya kalau yang bukan GMO. Kalau you tengok yang betul-betul cantik, betul-betul licin, tak bergerutu, semua ni, this is actually a GMO punya tomato lah. Okay? Ada yang kata GMO ni very bad. Ada yang kata is good. I have to agree it's both. Okay? Tapi banyak benefit lagi sebenarnya because when it comes to crops and also livestock, dia akan improve the quality. Cuma dia punya disadvantage maybe dia akan ada side effects to humans lah yang consume. Contoh macam human tu can get allergic reaction ke, atau yang betul-betul memang body dia tak tahan dengan GMO products. Some of the organs tu akan tak function efficiently ataupun can lead to failure of the functioning of the organs lah. Yang tu yang tahap kritikal yang teruk lah. So kalau just basic and your body boleh tahan, selalunya dia tak ada have any significant punya side effect sangat on GMO ni. Cuma nak kata long term tu scientists still finding out lah. Sebab dia kan kalau GMO kita bawa sequence baru, kita deal dengan dia punya DNA level dia molecular level, kita ubah the gene, the DNA. So scientists still trying to find out the serious side effect kalau dalam masa berapa tahun. It won't affect immediately sebab bila kita consume tu, badan kita still ada all this, kita ada immune system kita, and then kita also ada DNA polymerase yang akan repair ataupun try to correct the errors. But if it happens, then tak boleh nak ni lah. So scientist is trying to find out lagi what is the macam side effect yang kita boleh kata bukan jadi untuk seorang, maybe a pool atau dalam community, then we can call that as a side effect of GMO. Okay, so biotechnology has made impact in improving the human health, mostly in agricultural field lah. Dia akan produce increase the supply to increase the productivity in the agriculture yield. Also for food, nutrition and water security, and also to develop alternative fuel. Sebab we are relying now on petroleum fuel kan, our fossils are decreasing. So kita kena guna alternative fuel juga. Okay, to lessen the dependent on the petroleum fuels. Okay, these are the fields of biotechnology. Yang ni, this is not the latest. This is 2015 tak silap saya ni. Kalau you tengok now punya field of biotechnology, color dia ni semua dah jadi macam rainbow. Sebab all the nanotechnology pun semua dia masukkan jadi color juga. So what does all these colors differentiate? Actually, De La Vega, dia decide sebab terlalu broad sangat this field of biotech, terlalu banyak dia punya classification and everything, so they have decided to use a color code. This is UNESCO yang keluarkan the color code ni. These color codes have been used to differentiate the main areas of research. And then if you can see bawah ni, some colors describe wide and well-developed range of activity. Macam yang dekat atas ni, the blue and the green, you nampak banyak. Okay, is wide and well-developed. Kalau yang macam kuning ni, only concentrate on food, nutritional science, and nutraceuticals. So kalau you tengok blue, banyak dia punya ni, but mainly on life under the sea lah. Maksudnya melibatkan sustainable development goal yang melibatkan water. Okay, so blue ni ada aquaculture, ada fish health, ada nutrition, ada cloning of ada cloning and genetic modification, selalunya dalam breeding of the aquaculture punya organisms, and then ada fisheries pest and disease control. Okay, so kalau agriculture lagi besar, agriculture dia banyak. So some of it contohnya biofertilizers, agrobiochemical, preservation, and cloning, all that are all in under the green code. Kalau grey is more on the bioprocess industry. Okay, classical fermentation and bioprocess technology. Actually grey ni dia ada dua. Ni grey ni untuk khas bahagian fermentation. Kalau bahagian lagi satu industri dia color putih. Okay, white biotechnology. Kalau red biotechnology is only concentrating on medical. Okay, and yellow is the nutritional biotechnology. Okay, so the first one we have the marine biotechnology is blue. Okay, blue biotechnology is the marine biotechnology. So what is marine biotechnology? Is the application of molecular biological method to marine and freshwater organism to develop useful products and application. So kalau marine biotechnology setakat ni apa yang kita dah ada applications is for food supply. Okay, this is satisfy the growing demand for high quality products from fisheries and aquaculture. So food supply ni, however, kita kena memang betul-betul take into account, especially on food security. Sebab kita punya supplies for aquaculture ni is decreasing every single year. Makin lama makin the tons produced, maksudnya the metric tons produced for aquaculture and freshwater industry is decreasing tiap-tiap tahun. Kita tahu, kita nampak senang je kita pergi market, kita beli and everything, but the supplies are decreasing, so we have to find other alternative juga. How to improve the productivity, how to make it disease resistant, how to improve the growth, okay? And mungkin ada yang start dah nak buat transgenic fish, introduce fish, ada yang still nak conserve. So those are all under food supply of marine biotech. Okay, the second one is energy. Ada yang guna sekarang because of kita nak try guna less, nak varietykan the source of energy. They have produced biofuel. Biofuel ni banyak dia punya background dia kalau you baca. Ada yang from microalgae, ada yang from other sources, tapi yang kalau bawah marine biotechnology, dia selalunya dia akan guna microalgae or macro or microalgae. Okay, macroalgae untuk biofuel, and a normal oil yang lain dia guna microalgae. Okay, yang ni salah satu contoh lagi lah that you can read on. Another one is environment. Biosensing technologies for marine environment monitor. Sebab kalau you perasan, kalau dah dekat laut, dekat sea, dia selalunya banyak kali in a year dia akan contaminate. sama ada from spillage of oil ke ataupun from algae bloom ke apa, dia memang selalu akan ada contaminant. So they use this bacteria selalunya ataupun the component in the algae itself, dia akan guna untuk biosensing technology. Biosensing technology dia sangat broad juga. Dia tak spesifikkan on one or two issues that can only monitor. Dia banyak issues involved, but as long as you know they can the application of marine biotechnology, some of it boleh guna sebagai biosensing technology is enough. Okay, selalunya biosensing technology dia akan monitor contaminant ataupun pollution control in the water. Okay, so that is for the environment. Okay, for the fourth one we have the industrial products. Contohnya collagen, gelatin, spirulina, all that nak kata to boost our health atau boost our contohnya boost our immune system, macam itulah. Okay, those are production of marine biopolymers. Selalunya dia akan ada dalam food and also our cosmetics. Okay, so that is marine biotechnology. Okay, next we have agricultural biotechnology. Okay, agricultural biotechnology is the range of tools that alters the living organisms to make or modify products, also to improve the plants or animals, or also to develop microorganisms for agricultural use. Okay, sama juga lebih kurang macam marine. Okay, so actually for food supply and security, dia nak increase higher yielding in crops and livestock. Okay, kebanyakan that we know kalau agriculture biotechnology dia akan involve genetically modified organism, sama ada dalam bentuk transgenic ataupun dalam bentuk recombinant DNA technology. Okay, contoh yang kita ada, yang kita tahu sekarang ni kebanyakannya corn, ada tempe kan, ada beans, ada potatoes, macam tomato tadi. Okay, all that is to enhance nutrient composition, also to improve the quality and also make it resistant to pests and disease, supaya kita tak rely on pesticide. Okay, dia insertkan the gene into the corn atau into the potato to make it resistant to pests and also to disease. Okay, next one we have the biopesticide and biofertilizer. So pesticide kan memang harmful for the environment. So dia ada alternative. We have the biopesticide and also biofertilizers, which is to manage the pests and also pathogen and weed. Contohnya the latest one that they can they come up adalah biofertilizer atau biopesticide guna baking soda. Dia ada chemical dalam baking soda tu yang insect tak suka. Okay, another one is the canola oil. Okay, lagi satu untuk fertilizer, dia guna rhizobium which can fix the atmospheric nitrogen in the soil. Okay, that is for biopesticide and biofertilizers. Last one is the nutraceuticals ataupun medicine or nutritionally functional food. Contoh yang akan bagi kita antioxidant, the dietary supplement, dairy products, citrus fruit, vitamin. Okay, all that are nutraceuticals. Okay, next one, we have the industrial biotechnology. So industrial biotechnology focuses on the production and processing of the chemical. Okay, bila you dengar industrial, selalunya dia akan melibatkan chemical, materials and energy using. Ni, tiga benda ni, chemical, materials and energy. Sama ada guna yeast, guna fungi, guna bacteria, plants and also enzyme. And dia akan produce massively in a mass production untuk industrial scale synthesis of product. Okay. Industrial biotechnology memang dia deal dengan mass production. So when we say mass production, the first one is we have the development of new sustainable energy sources, bioenergy and biofuel. Okay. Kalau you realize that some of the application ni overlap, yes, betul. Dia akan certain macam biofuel atau macam dalam GMO, dia akan overlap dalam each of the field tu because they use the application. Okay. So industrial biotechnology is bioethanol. We have the bioethanol and also the biodiesel now as a choice other than petroleum. And then ada dalam food additive. Okay, dalam kita punya sweeteners and everything. Okay. Ni selalunya ada dalam food, pharmaceutical and also cosmetic. Perfume kita pun semua, that is all under industrial biotechnology. Okay, this one down here is the production of chemical ingredient yang selalunya kita guna dekat lab tu ke, ataupun memang for massive lab production yang the government guna. So these are producing bulk chemicals. Okay, so selalunya organic acids atau alcohol which contribute to energy saving, gas emission reduction and also assurance of industrial innovation and supplies. So what you have to know ialah chemical ingredients saja. Okay, dia memang produce in bulk. Okay, the last one is design and production of new biodegradable plastic textile. Okay, kita nak sekarang semua nak go green and everything. So kita nak guna less of the normal plastic tu because dia take a very long time to degrade. So sekarang kita bertukar kepada biopolyesters which can be fully biodegradable. And they release water and carbon dioxide into the environment. So kalau dalam kedai atau kedai mana-mana you pergi still guna plastik yang lama tu, kalau boleh you elak. Okay, you letak dalam your handbag ke, atau you just carry it. So kita nak try to apply the Go Green punya campaign, which is to say no to plastic bags and then say yes to biodegradable plastics. Okay, so that's industrial. Next, we go on to the red biotechnology, which is the medical. Okay, medical biotechnology is the use of organisms and organisms-derived materials for research and to produce diagnostic and therapeutic products that help to treat and prevent the human disease. Okay, kalau yang ni, this is very common lah. I think you all know drug discovery memang producing of new drugs and everything. And the latest one, nak kata latest tak sangat lah. Few years back, dia guna pharmacogenomic testing on colorectal cancer and leukemia. Maksudnya dia produce the drug ni yang that person ataupun that patient boleh adapt in terms of the genetic makeup. They try at the molecular level. This is new drugs discovery. Okay, the next one is gene therapy. Gene therapy is to treat the disease by replacing. Yang tadi dia introduce new drug into the genetic makeup. For gene therapy, it replace defective gene. Okay, the replace defective gene ni with a healthy one. Contoh they are trying to do for cancer. Cancer sangat-sangat susah sebab dia sangat rapid dia punya division. But they tried for Parkinson, HIV and influenza. HIV ada berkesan juga sikit, maksudnya dia prolong the longevity of the patient lah. Okay. So the next one is the stem cell. Okay, stem cell ni yang selalu kita guna sekarang lah, yang what kita trying to implement. Tetapi nak kata it is widely applied in Malaysia, not yet lah. Tapi memang is widely applied dekat the western countries, memang dorang guna stem cells already. Stem cells are introduced into a damaged area of the body which replace the damaged tissue cell. Selalunya damaged tissue cell ni can be anything, tapi yang kita selalunya buat ialah kalau ada orang tu dah terlibat dalam accident ke apa ke, dia dah injured, contohnya dia punya spinal cord atau apa, so we try to fix that using the stem cell. Kita buang the damaged tissue cell, kita akan replace with the new one. So yang ni stroke pun ada juga. Kita inject modified stem cell directly into the brain after a stroke. Maksudnya tu pun dia buat keluar the blood clot. They try to inject modified stem cell kalau the patient perlukan. Kalau buang the blood clot, the patient can still recover, then they won't insert the stem cell. Okay, so the last one is tissue engineering, which is the creation of the artificial. Sekarang banyak artificial kan, cosmetic and all. So yang ni kalau dalam medical punya term, dia akan guna untuk artificial skin, artificial cartilage and also artificial bone marrow. Ini selalunya kita akan treat patient yang ada burn and also severe injuries. Okay. So next one is food biotechnology. Okay, food yang tadi kalau you tengok dia berkait rapat dengan agricultural punya biotechnology as well. So food biotechnology is the production or processing of the raw food material using biotechnology approach. Dia selalunya melibatkan enzim, microbial ataupun GMO. Okay, So what is GM atau genetically modified organism or genetically modified food in this term of food biotechnology? So animals, plants and microorganisms that contain modified genes to improve production yield, quality and also the nutrition related property. So food and ingredients that are prepared from GM plants, animals and microorganisms, contoh macam tofu, they have tempeh, there is soy-based milk, soya bean, there is soy flour, tomato paste. Tomato paste ni yang kita selalu guna buat pasta tu is actually by GMO lah. Okay, and then sauce lah selalunya, sauce tomato, sauce chili, sauce semua tu lah. Selalunya dia akan ada element of genetically modified. So another one is food that contain molecules that produced by GMO that provide the health benefit beyond the basic nutrition. Contoh macam yang very famous is the golden rice. Okay, rice kita selalunya putih kan? Tapi kalau rice dalam ni, kalau you perasan dalam gambar ni, transparentnya gambar, this is the golden rice. Okay, the yellow. So this golden rice has received genes from a daffodil. Daffodil kan color yellow. So, and a bacteria that enable it to make beta carotene. So that's why dia jadi yellow. So yang ni kalau selalunya golden rice ni, they claim that it has the additional nutritional value compared to the white rice. Okay. Okay, so next we go on into environmental biotechnology. It involves solving environmental problems through the application of biotech. Contoh biomarker. Biomarker are responses of an organism which indicate the exposure of a toxic effect. Biomarker ni toxic effect, okay? They detect the levels of toxin in the ecosystem ataupun they detect airborne pathogen. Contohnya macam anthrax. So we have a specific biomarker that scientists play with lah. Setakat tahap mana yang safe and mana apa akan jadi kalau it becomes not safe. So diorang try to fix a certain amount atau a certain level of toxin punya reading ni untuk jadikan sebagai biomarkers. Okay. So next one is the biosensors. Biosensor is the analytical device to detect the sensitive biological element. Okay, yang ni saya tak berapa nak familiar with, tapi this one dia guna arsenic. Arsenic ni tak silap is a compound yang apa ni? It's in the air, tak silap saya. Nanti I'll try to read on this. Arsenic biosensor to detect arsenic use the arsenic operon of the bacteria. Okay, this one I'm not that sure how to elaborate on. So kalau siapa-siapa tahu, you can add. Tapi try to just read on biosensoring in the environmental punya biotech. Okay. The next one is the biofuel again. Sama juga lagi, biofuel or bioenergy derived from biomass from plant or animal. Macam you tengok dalam ni, they are trying to use corn. Okay, corn stover ini dalam bioethanol. And then last one is the bioremediation. Okay, yang ni selalunya important kalau when it comes to environmental biotechnology. Soalnya when you mention environmental biotechnology, you belajar pasal apa? Selalunya you akan concentrate more on bioremediation, which is the process of cleaning up the hazardous substance into non-toxic compound using biological organism. Contoh, selalunya dekat laut ke dekat sungai ke apa, kita akan selalu ada non-toxic compound because of human activity. So because of human activity, contohnya macam oil spillage ataupun chemical ke dibuang dalam laut ke semua tu kan. We use bacteria selalunya. Bacteria because dia punya apa ni, characteristic that can clean up the ocean oil spill ataupun to decrease the level of contaminants in the sea or in the river. Okay, this is called bioremediation, maksudnya dia cleaning up. When you hear bioremediation, you kena ingat is the process of cleaning. Okay, dan selalunya kita akan guna bacteria. Okay, next we go on into omics technology. Omics. Okay, omics ni apa? Omics ni all this at the back is the application of tools to learn all this. Mix. You perasan kat belakang ni semua ada mix. So when it comes to DNA, at the DNA level is called genomics. Okay, this one you have to really know. You have to understand apa beza antara. Not into detail sangat, but you have to know the beza. the perbezaan. Selalunya memang akan ada soalan ni. Okay, genomic is for DNA. Okay, it is to understand the entire genetic information content of an organism. So that's why scientists study genomics at DNA level. Kalau dekat mRNA level, it is called transcriptomic. Selalunya siapa yang buat transcriptomic macam saya buat transcriptomic untuk my PhD, I deal with expression. So, it is the study of RNA molecule that are produced by the genome. It is essential for interpreting the functional elements of the genome. Daripada DNA, kita ada mRNA. Tapi selalunya saintis, kita ramai saintis. So, we have to actually concentrate on a certain field. Dalam contoh saya dalam aquaculture, kita akan develop new sequence atau new biomarkers ke, tengoklah apa yang ikut your field. Tapi if dia melibatkan mRNA, dia akan fall under transcriptomic. You deal with mRNA sequence ataupun you extract the mRNA and everything. That is called transcriptomics. Okay, it's the study of the RNA molecules that are produced by the genome. Okay, next one is proteomics. Selalu yang ni pun selalu dengarkan proteomics. Proteomics deal dengan protein. Kalau protein is the study of structure, function and interaction of cellular protein. So kalau goal of the proteomic ni actually to identify and also to catalog every protein in the biological system. They try to, tapi nak produce for every single organism in the biological system will take a very, very long time, and sometimes orang kata it is not possible. But they still put it as the goal of proteomics. Okay. Proteomics is at the protein level. Okay, protein, proteomics. Okay. Last one is the metabolite. Okay, metabo. Say that's the keyword. Ada punya tips for you to remember. Metabolite, metabolomics. Okay, metabolomics. Selalunya jarang sangat-sangat, selalunya dalam bidang ni dia tak berapa nak as widen as the genomic and the transcriptomic punya field lah. Metabolomic ni tak berkembang sangat lagi. Dia guna metabolite. Okay, the study of metabolite in a given cell, tissue or organism. Selalunya dia akan berkaitan dengan biochemistry. So siapa-siapa yang berminat nak masuk ke biochem, dia akan banyak belajar on metabolomics. Okay. Okay, so these are the application of omics technology dalam the current situation sekarang lah. So kalau genomic, we have the Human Genome Project. Human Genome Project. Yang ni dia bukan boleh buat dalam masa setahun atau dua. It takes years to complete the Human Genome Project. Okay, apa yang dia buat is to identify around 20,000 to 25,000 genes in the human DNA. Why lama sangat? Sebab zaman dulu dia dah start daripada 90s. Dia sekarang punya teknologi makin cepat. Maksudnya you can get the result. Contoh kalau zaman dulu, it will take 3 months to get up to 100 punya gene. Tapi zaman sekarang, 1 month you dah boleh dapat 100 gene. Yang tu pun kalau you work in a team lah. Okay, tapi I'm comparing the technology now. We have the new generation sequencing, we have a lot of the Illumina sequencing, banyak yang dah upgrade daripada zaman dulu. So now we can do more projects lagi cepat. Okay, so the Human Genome Project ni determine the sequences of the 3 billion base pair that make up the human DNA. 3 billion. Okay, that is genomics. Kalau transcriptomics is the biomarker discoveries of diseases. So transcriptome of healthy and cancer cells can be studied by researchers to understand the what causes cancer ke, everything you deal with the expression. Macam saya for my PhD study, I did on behavior and also color changes. So that is expression kan, the traits. So saya cari genes yang berkaitan dengan color changes lah, and also for aggressive behavior. Okay, so this one next is the proteomics. Proteomics selalunya melibatkan food industry, which is protein, kan, kita deal dengan protein. So proteomic technology can monitor and characterize the protein content in the food and also to help to identify the quality protein biomarker for validation of food security in the industry. So ni proteomics. Okay, the last one tadi is the metabolomics. Is the drug discovery. Sebab dia biochem kan, dia deal with all the chemical component. So selalunya dia akan deal dengan drug discovery. It can monitor treatment efficiency of drug for certain disease at a very early stage. Okay, and also provided detailed view about the biochemical or physiological status changes caused by drug. Selalunya metabolomics ni punya field, dia akan berkait rapat dengan hospital. Dia akan bekerjasama dengan hospital and also pharmacist. Okay, yang lain ni ada yang boleh setakat research in the institute ataupun dalam universiti pun boleh buat juga. Tapi selalunya metabolomics dia walaupun dia buat dekat universiti, tapi dia akan berkait rapat atau akan ada jaringan dengan industri. Contohnya macam hospital and pharmacist. Okay. So okay, this is the Human Genome Project. Kalau you tengok the number of years dia take untuk nak sequence the DNA genome, dia take from 1990 sampai 2003. Tapi yang ni pun belum actually complete. They declared it as complete because they published it. Tapi ada a new, about 2 years ago, dia orang kata it is not complete. Because bila now we have all the modern technologies, bila dia check balik, dia kata it is still not enough. Tapi not enough tu pun bagi saya dah banyak, dah dapat sampai 20,000, 25,000. Kita kalau kita nak buat vaksin ke apa, we don't guna sampai banyak macam tu, but at least we have the data. Okay, so they started off the meeting and everything dekat before the 1990s, and then in 1990 it officially began. Okay, the Human Genome Project sampai ke 2001, and then 2003 dia betul-betul stop the project, maksudnya there's no more funding. Okay, right. So daripada the genomics and also transcriptomic, selalunya kita akan buat the bioinformatic. So kalau genomic dengan the proteomic, kita akan cari dia punya, kita akan buat dia punya sequencing. So in order to read the sequence semua tu, kita akan guna bioinformatic. Bioinformatic ni is actually the application of computer technology, specially developed software ataupun statistical technique to study the biological information dalam genomic and also proteomic. Kalau you setakat buat genomic saja, you tahu you just buat dia punya you target dia punya gene ke apa, you tak tahu the in detail lagi what is the function of a certain gene tu. Maksudnya you tak boleh nak blast dia. Contohnya kalau you buat genomic untuk benda baru, contoh dalam kumbang you nak tengok DNA dalam kumbang. Tak pernah ada orang buat lagi DNA dalam kumbang. You dah pergi sequence, you dah hantar ke lab, you dah dapat the sequence semua. Tiba-tiba, you dapat information tu je. Macam mana you nak tahu what is, apa relation dia dengan insect? Kumbang ni betul ke dia punya sequence ni related dengan insect ke macam mana? So this is when it comes to bioinformatic. Kita akan process the information tu. Whatever sequence atau whatever you get from genomic and proteomic ni, kita akan selalunya kita proses melalui the bioinformatic. From this bioinformatic baru you tahu berapa banyak gene dalam kumbang tu. How is it functional? Berapa yang functional dalam producing the kumbang tu punya shell color, dia punya color dia tu. Ataupun berapa banyak yang produce the eye color. So you can know from bioinformatics. Okay, so from this bioinformatics juga you can also have information how to alter the DNA. Okay, so that's this bioinformatics sangat-sangat becoming an important field lah sekarang. But dia sangat-sangat challenging. Saya buat saya punya PhD transcriptomic and then I use a bit of the bioinformatics pun dah sangat-sangat pening, tapi those I don't encourage, I don't discourage you to take bioinformatics. It is very good nanti for future, especially all this vaccine and all the penyakit dekat ni kan. So it is very good if you can venture into bioinformatics. Okay, bioinformatics will become an important field nanti dalam masa depan kita insyaAllah. Okay, sebab kita ada banyak new technologies in genomics and also proteomics. The application or the use of this bioinformatics is very, 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 very important. Okay, Selalunya bioinformatic punya application ni selalunya kita akan guna untuk develop all this. Okay, personalized medicine, gene therapy, drug development, antibiotic resistance, evolutionary studies, waste cleanup, biotech generally, okay, microbial genome, molecular medicine. All these are under genomics and also proteomics. Okay, sometimes you can also use it macam saya, transcriptomics saya guna juga. Tapi kalau in general term, you can use it for genomic and proteomics. Okay. Alright, next one. Okay, don't get confused with the metabolomic yang tadi tu. Metabolomic is the metabolite. This is metagenomics. Lain. Is the environment. Kalau you tengok metagenomics, tak nampak pula. Okay, metagenomics selalu berkaitan dengan environment. Is the genetic materials obtained directly from the environmental samples. Okay, is to determine the evolutionary interaction in a particular environment by revealing the hidden biodiversity of microscopic life. Okay, dalam ni kalau kita ambil from the environment, contohnya daripada space ke kita bawa balik, daripada soil, daripada water, daripada tissues, okay, these are all metagenomic. Okay, kita balik, kita dah buat sampling, we analyze. Ini you tak payah tahu, okay, dia detail. Ini saja je orang letak ni. Ada gambar. Just to for you to visualize. You have to know metagenomic ni apa. Okay. So when it comes to the analysis, you have to cut the DNA. Lepas tu kita akan buat shotgun method and then we do the profiling. Which is dia akan melibatkan also some part of bioinformatics. Not all. Okay. Cuma some part of it is the blasting of the sequence lah. Okay. So this is metagenomic. Meta is the environment. Metabolomic is the metabolite. Okay, don't get confused between these two. Okay, next we go on into the important part, which is the techniques in biotechnology. Okay, the first one we have the recombinant DNA technology. Recombinant DNA technology is the joining of the DNA molecules from different organisms. Okay, RNA, recombinant DNA, where we call it as the rDNA, is inserted into a host organism to produce new genetic combination and product. Okay, contohnya yang ni, DNA from other sources. Okay, this is the gene sequence. This can either be original sequence daripada dalam DNA dia ataupun can be engineered sequence, depending on kita punya experiment, kita nak buat macam mana. So the gene ni is cloned into a vector. So this is the bacterial plasmid, we call it as the vector. Okay, kita masukkan the foreign gene sequence. So this is now called the recombinant DNA, rDNA. Okay, sebelum ni asal dia merah je, tapi kita introduce the blue. So that's why it's called recombinant DNA. Okay. Another method, recombinant DNA selalunya melibatkan DNA cloning juga. This is part of the technique used. DNA cloning ni is making identical copies of DNA in a large quantity. Selalunya when it comes to DNA cloning, memang kita hanya akan make identical copies in a very large quantity. Okay, cloning sama je dia akan jadi identical copies. So this is the analogy dia, Star Wars. Okay. Requirement. Okay, this is what you have to know. Requirement of DNA cloning. Dia mesti ada gene of interest. Okay, selalunya kita akan amplify dia guna PCR machine. Okay, another one is dia kena ada restriction enzyme yang akan potongkan the DNA, masukkan the vector. Okay, and then we have the vector which is the plasmid. Oh sorry, yang tadi restriction enzyme untuk masukkan gene. Sorry, not the masukkan the vector. Masukkan the gene. So the vector is the plasmid itself. Ini. And then we have the DNA ligase. Ligase ni tak ada yang tanya, kenapa ada dalam DNA yang hari tu kita belajar, yang ni pun ada lagi. The DNA ligase ni actually function dia memang untuk join DNA fragments. Dia macam glue. DNA ligase ni you have to remember that is a UHU glue. For in nak buat the, janganlah cakap dia UHU glue, but you visualize it as a UHU glue. Okay, it joins the DNA fragments together. So DNA ligase ni is to join, insert and vector. And then we have the host cell balik, copies the clone DNA using its own replication mechanism. So kalau you tengok dalam ni, okay, this is the bacteria and the plasmid yang dalam bacteria ni. Okay, kita ada gene of interest. So gene of interest kita, contohnya kita nak color ke apa ke kita nak masukkan dalam this bacteria. So kita ada gene of interest, take out the gene dia, the gene is inserted dalam plasmid. Ini. Okay, gene dimasukkan ke dalam plasmid. So now we have the recombinant DNA or the recombinant plasmid. So plasmid ni akan put into the bacterial cell balik. So now the bacteria has become recombinant bacteria. So dia akan replicate sendiri. Okay? That is DNA cloning. Memang it makes identical copies of DNA in a large quantity. Okay, next is gene editing. Gene editing is usually kita akan cakap dia selalu turn on and turn off. Contohnya is usually melibatkan addition and deletion. So making a small controlled change in the DNA which allows the genetic material to be added, removed or altered at a particular location in the genome. So the famous method is the CRISPR method, we call it. Okay, CRISPR. Tak payah you hafal this cluster, regular, blah, 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 blah. Dia akan CRISPR. Okay, CRISPR or Cas9. So if you see this gambar here, dia potong buat keluar the piece of the sequence and they insert yang baru. Okay, this is called editing. The cut and edit. So DNA editing ni, dalam ni contohnya, okay, specially design punya synthetic guide molecule, dia cari the target DNA strand. Okay, kita cut this thing off, kita masukkan dia dengan healthy DNA strand. Okay, so this is the defective one. Cut the defective strand, kita masukkan the healthy strand. Okay, selalunya akan melibatkan the removal, the removal and also the addition. Okay. Okay, another one is gene therapy. Gene therapy ni dia ada dua, ex vivo and also in vivo. So a technique that use genes to repair faulty gene for treatment of disorders and various human illness. So gene therapy ni dia akan sama ada buat luar, ex vivo which is outside the body, and another one is in vivo which is inside the body. Selalunya in vivo kita akan cucuk terus masuk ke dalam badan. Kalau ex vivo kita akan buat keluar contohnya darah, buat keluar dulu darah tu, and then kita treat dia, lepas tu baru masukkan balik darah tu ke dalam badan manusia. So that is ex vivo. Kalau you nak baca in detail on ex vivo punya contoh and also in vivo ada dalam your medical textbook. Okay. So ex vivo berlaku dekat luar, outside the body, and in vivo berlaku dalam badan, dia terus cucuk masuk. Okay. Okay, last one, biotechnology product. Kita ada GMO in, first one we have the GMO lah. GMO yang macam tadi saya dah cakap banyak kali dah. Kalau dalam agriculture, GMO ni is to increase the crop yield per area, reduce use of chemical insecticide and everything. Okay, saya nak tanya ini, how do you differentiate between a transgenic animal atau transgenic punya plant dengan a GMO? Kalau transgenic, dia selalunya melibatkan DNA daripada lain inserted into a new DNA. Kalau GMO, dia tak melibatkan the outside selalunya lah, dia tak melibatkan the outside. Dia akan potong atau edit the gene, the sequence of DNA dalam the original organism sahaja. This is GMO. Okay? Dia tak melibatkan new gene. Tapi it can be confusing sebab dalam internet semua ada, tak tahulah, they don't know how to differentiate betul-betul GMO and transgenic. So dia buat, kalau you baca dekat internet, GMO ada yang insert tu, insert ni. Tapi the originality of GMO, dia tak melibatkan DNA baru masuk dalam badan dia. Maksudnya dia akan alter from what the DNA is produced in the body, sama ada dia delete ke ataupun dia buat gene editing lah, using genetic engineering. Yang kalau transgenic, dia akan introduce baru, recombinant. Okay. So selalunya transgenic ialah GMO punya, transgenic is part, macam mana cakap, transgenic involves GMO, but GMO sometimes does not involve transgenic. Okay, you have to know that. Okay, nah ni transgenic. Yang tadi, this is, dia kata GMO which have been inserted with DNA of different species. Okay, like I said, transgenic ni, dia melibatkan GMO, tapi GMO tak semestinya melibatkan transgenic. Okay, so GMO yang ni selalunya dalam animal, dalam plant, and also ada certain risk lah untuk nak katakan this transgenic animal and plant. Sebab kita contohnya, kita yang the famous one is the glowfish. Kalau you tengok, you google glowfish. Glowfish tu ikan zebrafish je. Tapi dia boleh glow, dia ada fluorescent color. Why? Because it has been recombinant dengan a jellyfish. Jellyfish yang ada fluorescent. So dia ambil DNA daripada the jellyfish tu, dia masukkan dalam the zebrafish and then the zebrafish to glow. Okay, that is called the glowfish. Okay, that is transgenic. Dia masukkan daripada jellyfish, masukkan kepada the zebrafish. Okay. So these are examples of transgenic ini, enhanced reproductive performance dia, increased growth rate, improved milk production or composition, and the implants longer shelf life. But there are risks because takut nanti kita manusia kan, kita selalu lupa kita in terms of ethics tu, maksudnya you dah boleh alter the organism, you ingat you are playing God. So that is one of the risk dia lah. And also the safety potential human health implication. Tu yang that one scientist tengah find out lagi. All these GMO semua ni, does it have betul-betul punya side effect other than those claim earlier? Yang claim earlier ni because of allergic reaction, because of mild side effects. Tapi they are still trying to find out whether it has severe side effect on the community. Okay. Okay, last we have the biocatalyst. So biocatalyst is the substance such as enzyme or hormone that activates or speed up a biochemical reaction. Okay, selalunya kita akan guna dalam cleaning agent. Okay, biocatalyst ni enzyme. Okay, biocatalyst usually melibatkan enzyme and also macam lipase selalunya. Okay. Dia akan, kalau yang application that we use biocatalyst now is the cleaning agent. Kita guna dalam food and beverage, betulnya dalam fermentation pun kita guna, dalam biofuel production, dalam medicine as well. Okay, kalau probiotic macam Vitagen yang macam Yakult semua tu, what is it? Is that a biocatalyst? Rasa-rasa tu biocatalyst ke tak? Siapa-siapa yang boleh jawab?
Dia orang guna bakteria.
Ya, betul. So it is not a biocatalyst. It's GMO kalau probiotic tu. Okay, so those are the type of question that you will get in your exam. So apa yang you faham and the overall punya macam nak cakap the overall techniques in biotechnology tu. Okay, so that is all for our lecture. So is there any questions at all?